and I'm going to talk about ocean acidification and coral reef fishes. Is it a threat to coral reef fishes? Just to start with, I'd like to thank the people that have really supported this work, the co-authors, Daniel Dixon, Jennifer Donaldson, Mark McCormick, Mark Meakin and Monica Gagliano, and a few people that have been heavily involved and other organisations supported the work. So thank you to those people. As Ove told us very clearly yesterday, about 30% of the excess carbon dioxide that's been produced by humans over the last 250 years has been absorbed by the ocean. And global warming would be worse if this wasn't happening. So, no, there's maybe a bit of a positive in that, but there is a cost because the ocean is becoming more acidic and it's changing the carbonate chemistry of the ocean. Now, I'm not going to go into the details of the equations here. It's enough to say that PCO2 levels are going up, carbonic acid, bicarbonate and hydrogen ions are increasing in the ocean, carbonate levels and pH are going down. And pH is estimated to have declined by about 0.1 of a unit since the start of the industrial age. If we look at where things are going, atmospheric CO2 is currently at about 390 parts per million in the atmosphere. It is likely to exceed 500 ppm by 2050, the way we're going at the moment, and on current trajectories we will be somewhere between 700 and 1,000 ppm by the end of the century if we don't do something fairly serious about changing the amount of carbon dioxide that's going up into the atmosphere. Now, this would lead to a pH decline of the ocean, somewhere of the range of 0.3, another 0.3 to 0.4 units. And this would be a real problem, of course, for marine calcifiers, such as corals and echinoderms and bivalves, because the concentrations of carbonate ions would decrease. It would be harder for them to actually make their skeletons. And aragonite saturation, for example, would go below 1 somewhere in the second half of the century. But there are other effects of increasing CO2 in the ocean as well. It's not just about calcification. High PCO2 in water can affect the acid-base balance of organisms. So, in fact, it can cause acidosis of the internal tissues. And in invertebrates, the sorts of things, there's been some work done that suggested that things like we're seeing reduced fertilisation success, reduced growth and survival, and abnormal development at lower pH levels, at higher PCO2 levels. And this is particularly in the early life history stages, in the larvae, the eggs, and the likely reason for this is that these very small life stages have a larger surface area to volume ratio, so they're more susceptible to the external environment. And the other thing is that they have reduced, or they don't have as well developed, acid-base control mechanisms at that stage. So it's likely that these early life history stages are among the most susceptible. What about fish? What are they like at regulating their acid-base balance? Well, it turns out that adult fish in general, from what we know, are probably pretty good at that. They can buffer their tissues with intra- and extracellular buffering with, for example, bicarbonate. They can exchange ions across the gills, and so they can control their pH pretty well. But we do know that very high PCO2 levels can affect survival, growth, development and metabolism. It's pretty well recorded, but we don't know what the effects might be at levels relevant to the climate change issues that we're really interested in, the sorts of levels I just told you about a couple of slides ago. And if there are any effects, we would expect that they would be most marked in the egg and larval stages, in these early life history stages. So that's what we've been doing. We've been looking at the effects of elevated CO2, reduced pH on the early life history development of a couple of model species. First of all, good old Nemo, Amphiprion percula. This has been one of our model species, and the other one is one that we've heard about today as well from Chris Fulton, for example, the spiny damselfish, Acanthochromis polyacanthus. And we've been rearing these fish under a range of CO2 treatments for controls of 390, then a moderate level of around about 550 to 600, then higher levels of 700 to 750 or 850 to 1050. So a range of levels relevant to climate change predictions. And for the clownfish, we do this from the egg stage up to 11 days post-hatching. That's when they finish their larval stage and they're competent to settle. And for the damselfish, we do this for eggs up to 21 days as juveniles. And we have multiple parental lineages and lots of replication. And the sorts of things we're looking at in the early life history are things like embryonic survival, duration, egg survival, size at hatching, growth rate, skeletal development, swimming performance and larval behaviour. Now, I'm not going to spend lots of time detailing the results here. I just want to tell you there's some pretty good news in terms of early life history development. For these two fish so far, we've seen no negative effects of elevated CO2 on embryonic duration, hatching success, growth, larval survival or swimming ability. Now, there's probably a lot more work that needs to be done, but most of the things that we've looked at so far, we're not seeing very serious negative effects on those sorts of traits. So that's some pretty good news. It looks like these guys might be able to handle mild increases in CO2 in terms of these traits. And even with the skeleton and the otoliths, things are looking pretty good. We've looked at how the skeleton develops and we see no effect of elevated CO2 on the skeleton. And even more importantly, we see no effects so far on the otoliths. And the otoliths are particularly interesting because they're paired ear stones. They're made of aragonite, so we might suspect that they'd be quite susceptible to changes in carbonate in the seawater, as reduced carbonate concentrations in the seawater, or because of acid-base control in the fish themselves if they're changing the carbonate bicarbonate levels in their tissues. But so far we can't see any effects of changing the pH and the CO2 levels on the size, the shape or the symmetry of the otoliths, and these are all things that we've heard about today as being quite important. But it's not all good news. As you may have read recently, we've actually found some negative effects on the olfactory ability of these fish. So if we rear them at around about 1,000 parts per million CO2, reasonably high level, but not outside the realms of where we're going, there's an impaired ability of these larvae to discriminate between ecologically important chemical cues. So they can no longer discriminate between the smell of different types of settlement habitat, between kin and non-kin, and between predators and non-predators. And these are all things that fish reared in control conditions can do really well, but at elevated CO2 they cannot. And of course you can imagine that losing these sorts of really critical functions at settlement could have a big effect for recruitment and the success of populations. So we've been looking at that. We want to now know what sort of concentrations below 1,000 do the larvae actually get affected. Do more species than just Amphiprion get affected? What other behaviours are affected? And does this actually lead to an increase in mortality in the real world? We've been rearing the fish either in captivity or we catch them in light traps at the end of their larval phase, and that's Mark McCormick up at Lizard Island setting a light trap. Then we can rear these little fish with elevated CO2, either at these control levels or a range of elevated CO2 levels that are there on the right of the slide. And we can then test their preference for a whole range of smells. And we can do this in a mini flume chamber where we have two different sources of water that come down different channels of the chamber, and then we can put a little Nemo in the centre of the chamber and see which side of the chamber it prefers. We can swap the water sources and retest to control for any side effects. And this is the sorts of results that we get. And what I'm showing you here is the preference of the larvae for a stream of water containing the odour of a predator, a little rock cod predator. And this is for every day from hatching through to settlement. And this, first of all, we've got the control line and the 550 line. So these fish reared at control water or 550 water. You see that they strongly avoid the smell of the predator, and then by about day 8 they're avoiding it altogether. So they do not like the smell of a predator. That's pretty good news for them. When the fish are reared in 700 ppm seawater, though, we start to get a different result. And after about 4 days, what we see is that they start to increase the use of the side of the chamber that has the smell of the predator in it, and it's up to about 50% on average. But more importantly, you'll see there's these very big standard deviations here. And what this is because there was a lot of variation between individuals. So at this level, what we see is that some individuals continue to avoid the water stream containing the predator odour altogether. Others start to become strongly attracted. So we have more or less this dichotomy of results, of responses, where some individuals are strongly attracted, others are still repelled. If we go up to a higher level, up to 850, it changes dramatically. We see this flip at about day 2, where the fish go from absolutely trying to avoid the water stream. In fact, at 850, we show they have very, very high attraction to that water stream. They spend most of their time in it. And you'll notice the small error bars there. There's not much variation among individuals. They're all doing the same thing. Now, this tells us a few really important things. It tells us that the time to induce an effect of CO2 is dose-dependent. It tells us that probably around about 700 ppm CO2 is roughly a threshold for this species. All the larvae are strongly affected at 850 ppm CO2, but it does suggest that there's some potential, some pretty important potential for selection at around about 700 ppm CO2, but probably not at 850, because at that stage all individuals seem to be doing very much the same thing and attracted to the odour. Then we wanted to know, well, that's Nemo, clownfish. What about other fish? And so we went out and we caught little damselfish in light traps, and then we treated them for 4 days, and we got basically exactly the same response. We found that the fish in the control and the 550 line avoided the cue of the predator all the time. After about 3 days in 700, 750 water, then we saw that we started to get an increase in the attraction to the odour of the predator and a lot of variation between individuals. At 850, after 2 days, they were all attracted. We want to know now, does this really matter in the real world? So we went out, we did a field experiment, we caught these larvae, we treated them for 4 days, then we placed them out individually on little coral heads, and then we monitored their behaviour in the presence of natural predators, and we monitored their survival over 30 hours. These sorts of things we can do with behaviour. We can measure things like the total distance that a little fish might move, the maximum distance that it could move, that it moves from shelter, the average distance it moves from the shelter, and then something we call boldness, which is related to how quickly they return to shelter and then how quickly they come out again after they've been disturbed. I'm just going to show you the results for the control and the highest treatment, the 850 treatment here, and what you can see is that these fish, that they actually moved further away, total distance that they moved was greater. They were a greater average distance from the shelter. They were a greater maximum distance from the shelter, and they actually had a greater boldness compared to controls. Of course, all of those things you might imagine would actually increase their risk of mortality slowly. And in fact, that's exactly what we saw, that the mortality rate of these little fish was 5 to 9 times higher if they've been exposed to elevated CO2 compared to controls. We saw no mortality at all in cage controls, and so that illustrates that the agent of mortality is almost certainly going to be predation. We do a very quick back-of-the-envelope sort of calculation and extrapolation, and this is an extrapolation, but I think it just illustrates the point. Now, this would lead to 90% loss of a settlement pulse in 1.5 days at 850 ppm CO2 and only a 50% loss in 10 days for the controls. We're now looking at a range of other possible issues related to CO2 exposure in fish, and it looks like their hearing is affected, their learning ability is affected, and there's some very general nervous system response that's affecting a whole range of senses. We're also finding that there's variation among species as well as a variation among individuals in relation to the CO2 levels, but clearly this is an area that needs quite a lot more work. So in conclusion, in terms of ocean acidification, we see no negative effects of elevated CO2 on early life history development and growth. That's great news. We see no effects on the otoliths, which is something that people predicted there'd be a lot of effects on, and we just don't see that. They are, however, exhibiting riskier behaviour and are unable to discriminate between ecologically important cues in the real world. This causes high mortality rates during a really critical life history transition and suggests that this could be a really important area for future research. And it also illustrates that there can be variation among individuals, and we need to consider how that would act as a selective agent and consider that when we're trying to calculate the implications for population replenishment. There, thank you.